Genetic research allows us to understand the causes of diseases, develop new treatments, and even trace our ancestry. Think of DNA as a vast library, with each gene being a book filled with stories written in the language of molecules. By reading these genetic stories, we gain insights into the fundamental processes that make life possible. Henrietta Lacks was an African-American woman whose cancer cells were the source of the HeLa cell line, the first immortalized human cell line and one of the most important cell lines in medical research. Her cells were taken without her knowledge or consent in 1951, and they have been used in countless scientific breakthroughs, including the development of the polio vaccine, cancer research, and AIDS treatment. Imagine the impact of one woman's cells dividing endlessly, contributing to the health and well-being of millions around the globe. But behind this scientific marvel lies a deeply human story. Henrietta was a mother, a wife, and a friend. Her life was filled with moments of joy, struggle, and resilience, much like any of us. Yet her legacy is intertwined with profound ethical questions. The story of Henrietta Lacks raises important issues about consent and the use of human tissues in research. Her cells were taken without her permission, sparking debates that continue to shape medical ethics to this day. Recent research has shown that it is possible to reveal a person's identity through sequencing, highlighting the need for policies to address the privacy of individuals and their relatives. As we celebrate the scientific achievements made possible by HeLa cells, it is crucial to also honor the woman behind them and to reflect on the lessons her story teaches us about respect, consent, and the human side of scientific progress, ensuring that privacy and ethical considerations are upheld in genome research. Studies have shown that a significant percentage of cell lines used in research are misidentified or contaminated, highlighting the urgent need for greater awareness and measures to prevent and detect contamination. This problem brought to light the critical need for cell line authentication. Ensuring that researchers are working with the correct cell lines is essential for the validity of their findings and the advancement of science. The problem of cell lines being contaminated by HeLa cells brought to light the critical need for cell line authentication. Ensuring that researchers are working with the correct cell lines is essential for the validity of their findings and the advancement of science. Cell line authentication involves verifying the genetic identity of a cell line, often using techniques like short tandem repeat or STR analysis. This process is vital for maintaining the integrity of research and ensuring that scientific studies are reproducible and reliable. Without proper authentication, researchers risk basing their work on false premises, which can lead to years of wasted effort and resources. As we venture further into the frontiers of scientific exploration, it is imperative to remember the lessons learned from the past. The story of HeLa cells serves as a stark reminder of the ethical responsibilities that accompany scientific progress. Moving forward, open communication, transparency, and respect for human dignity must be the guiding principles of scientific research. By adhering to these principles, we can create a future where science and ethics go hand in hand, fostering progress for the benefit of all humankind.